Hello student, I am Dr. Manish Kumar Gupta, Associate Professor, Department of Biotechnology, Faculty of Science, Veer Bahadur Singh, Purvanchal University, Jaunpur, Uttar Pradesh. Today we will discuss about the use of basic local alignment search tool through online server. In the earlier class, we had already discussed about the basics and fundamentals of basic local alignment search tool. So, how do you use web based BLAST? Suppose you are a first times user. So, either you can type the URL of the BLAST, that is a https blast.ncbi.nlm.nih.gov/blast.cgi, and if you do not know the URL, you can directly just search in a search engine such as Google or Bing NCBI BLAST. It will open the BLAST, the first page, and then click on the BLAST. There will be a portal will be open of the BLAST. In the BLAST, it will show the nucleotide, protein, BLASTX, T blocks, and different types of a BLAST options will be shown. You can install the BLAST at your system as a standalone, or either you can directly use on the web BLAST. There is a options called cloud based BLAST, where you can also execute the process on internet. It is known as a elastic BLAST, which is a useful application that can efficiently perform BLAST search for the user in the cloud. Demonstration that with two examples. At the same time, it hides much of the complexity of working in the cloud, lowering the threshold to move work to the cloud. So, as I had discussed just now, that one can download the latest executable BLAST versions. So, in this slide you can see the exe file and other files which can be installed on the Linux operating system and Windows operating system of exe files. As well as one can also download the databases required for this BLAST. In the BLAST database, you can go on the sites, you can click and you can download the databases on your local system also. What are the major BLAST program that is available on the web based BLAST? It is the BLAST-N, in which the query sequence is a nucleotide and the database sequence is also a nucleotide. The other BLAST is a BLAST-P, in which the query sequence is a protein and the database is also a protein database. BLAST-X is another program in which the query sequence is a 6 frame nucleotide sequence and the database is a protein, in which this nucleotide has to be translated. Another program is a T-BLAST-N, in which the query sequence is a protein and the nucleotide is the database sequence, which is a 6 frame. The other program is a T-BLAST-X, in which the query sequence is a 6 frame nucleotide sequence and the database is a 6 frame nucleotides. So, we had already seen in the BLAST 6 measures, which is depicted in the details for what purposes it is used. The other specialized BLAST which is used for sequence alignment is a PSI BLAST, position specific iterative BLAST, useful for protein profiles, query against protein database. PHI BLAST, which is a pattern hit initiated BLAST, useful for protein pattern against protein database. Mega BLAST, comparison of large set of long DNA sequences, and the other specialized program genomic BLAST, which is useful for complete genome alignment. The other important program is a primer BLAST, which is useful for designing PCR primers with primer theory. The next program is a RPS program, which is a reverse position specific BLAST, and it acts as a complementary and opposite to PSI BLAST by comparing your query to the PSSM position specific scoring matrix of the conserved domain databases. Next to that, the primer BLAST, which is useful for designing the primers using the primer 3 algorithm and checking their template specificity using BLAST against selected sequence collections. The next program is called Smart BLAST, which is useful for processing your protein query to present a concise summary of the 5 best protein match from well studied reference species in the landmark database. The other important specialized BLAST program is called immunoglobulin BLAST, searching for immunoglobulin or T cell receptor sequences against germline database for annotation of the input immunoglobulin sequences. The most important of the another program is VEC screen. This is useful for screening input nucleotide sequences against a library of known vector and other artificial sequences to identify contaminations. The next program is a CD search, which is a searching and protein sequence against a database of curated domains of functional analysis. This search is performed for all protein blast requests. CDART is another program which is useful for identifying conserved domains present in the input protein sequence, followed by finding other sequences containing these identified domains. The other program is multiple alignment, which is using constraint based protein multiple alignment tool, which is known as a COBALT, to align multiple protein sequences with search links available in all protein blast result page. The global alignment is another program in which NCBI's implementation of the Needleman's global pairwise alignment tool for the nucleotide or protein queries. MoleBlast is another specialized program which is useful for identifying neighbors for input nucleotide sequences from selected target database using BLAST, and then cluster the collections according to their sequence similarities using multiple alignment, that is a muscles. The next important program is a targeted loci, which is useful for searching against curated nucleotide sequence from bacterial and archaeal 16S ribosomal RNA or fungal 18S, 20S and ITS for species identification need. So, these are the different types of a program which we have seen. So, when we are using NCBI databases, there are different types of accession indications or prefix are used. One has to know about different types of indications of these accessions, what it indicates. For example, the accession prefix AC, it indicates the complete genomics molecules, usually alternate assembly. Similarly, there is another accession prefix called NC, which is a complete genomic molecule, usually reference assembly, and NG is a incomplete genomic regions. Similarly, NT, which represent the contigs or scaffold clone based or whole genome sequences. NW represent the contigs or scaffold of primarily whole genome sequence, and NZ represent the complete genome. Similarly, there is a huge list of accession prefix. One can go through this and they can find it out. There is a RefreSeq database section, which is a curated non-redundant set including genomic DNA contigs, messenger RNAs and proteins for known genes and entire chromosomes. There are another different types of a color indication symbols, which is a legend of sequence wear. One has understanding this about the color of this, which is used in the sequence viewer. For example, the gene is indicated in green color, RNA is indicated in purple color and conserved domains is indicated in a red color. Similarly, there are a multiple features and there are other symbols which are used, which can be seen in the slides, and one can go through this legend and they should be able to understand about the different colors. Whilst arrows inside features indicate the directions of the feature related to the top level sequence. Gray arrows in introns, for example, in RNA and coding regions features indicate the direction of splicing. So, we had already discussed about the basic features parameters of the BLAST in the earlier class. Once again today we are going to depict here through this slides how to use the BLAST2 sequence alignment. As you can see, once you go to the NCBI BLAST program, you will see there is a box will open. In the box you can paste your query sequence and there is an option called align to sequence. Once you are using this align to sequence, it will open the general parameters and other parameters which are important for the BLAST2 sequence alignment. You can see the gap, maximum values, word size, scores, gap penalty, gap cost, all kinds of parameters you can see in these slides. We had already discussed about in the earlier class. Once you are using this BLAST-N through web based servers, you will see there is a box will be open where you can paste your sequences in a FASTA file format, or either you can upload your sequence by downloading the sequence from NCBI database or if you are working in a laboratory or generating your own sequences, you can save that sequence in the FASTA file format and you can upload it, or directly you can paste in this box. The other things, if you know the accession numbers or gene ID numbers, you can also just paste that gene ID number, the database automatically identify that program. The other parameters like non-redundant databases and other parameters like which kinds of programs are going to be used when you are selecting the organisms, you can also select from the database. There are different indications of C, D, B and A has been depicted in this diagram, which can be useful for the user to go through these slides. So, these are the other parameters which are used for this BLAST, like we had already discussed in the earlier slide also, what is the maximum range of target sequences, the expected threshold value, word size, maximum match and other filtering parameters which is indicated in A, B and C in the slide, and based on that one can also select the highly similar sequences. If you are interested, you can go for the Mega BLAST and click on the BLAST options, it will start the processing of aligning the sequences. So, similarly, if you are having a query sequence of protein and your database is having a protein databases, then you select the BLAST P program. Earlier we had seen about the BLAST N, where the nucleotide is the query and the database is a query sequence. Here in the BLAST P, the similar box will be open, where you can paste your sequence in a FASTA file format, or you can upload your sequence through click uploading the sequence in a FASTA file format. Besides that, you can fix a query range from where to where you want to align the sequences. There is a right side box from where to you can select the position of a particular nucleotide and to where this can be aligned. And again you can select the what kind of databases you are going to use. It is a non-redundant protein databases you are going to use for this one. You can select it where the sequences are not repeated and then it can be selected. By default, most of the users uses the non-redundant databases and these are useful for aligning the protein sequences. There are different types of parameters, such as we had already seen in the case of a BLAST N. Similarly, here also in BLAST P, all the algorithm parameters are depicted in this slide. The general parameters are where you can see how many targets you are going to select in the beginning of this. The range of this target is 5000 from 100 to 5000. You can select 100, 10, 50, 250, 500, 1000. So, it is up to the user, and similarly one can put the value of word size from 2, 3, 5, 6. By default it is a 5. So, maximum match is also a parameters which can be selected by the user. Similarly, like algorithm parameter, there is a scoring parameter which kind of matrix the user is going to use, whether they are going to use a Blossom matrix or PAM matrix. All kind of links are given in the menu box in the matrix. By default, it is a Blossom 62 matrix which is the most widely used. So, if users or students are not aware what kind of matrix they should use, by default they can use the Blossom 62 matrix. Similarly, there is a gap cost penalty in which existence and extensions values are there. There is a menu drop options you can select as per your need, and similarly there is a compositional adjustment parameters are there, one can use that parameter also. This is for the BLAST P alignment. So, other parameters while going for the BLAST P, the algorithm is Quick BLAST P, it is a accelerated protein BLAST. The BLAST P is a protein BLAST, and Psi BLAST, position specific iterated BLAST, and pattern hit initiated BLAST and delta BLAST. Here we are using a BLAST P, that is why it is highlighted in a dot blue color, it is being selected. The filtering parameters and masking parameters are also very important parameter while going for the BLAST P. The filter parameter, it remove the low complexity regions and mask for lookup table only and mask for the lower case letters also. So, here you can see how once you process the BLAST, how the outcome will look like. There are different symbols are indicated A, B, C, D, E, F, all tells about the different story, and each one of them has been indicated in the slide, where A you can say it is a user login ID, where B tells about the each result is given in a row in the table with the identifier in the request ID. The C indicate the program, title, database columns combined to provide a summary for a specific search. The D indicate the database restrictions applied are indicated by more dot dot and the pop up upon hover D. The E option tells about to save the databases results from this BLAST. The save, download and read X allow saving the search strategy. The save strategy page shows M, J, K, L indicated, where one can save the results which are generated from this BLAST P. Here we are going to take one problem to use this BLAST. The problem is how do we identify the unknown protein sequence. So, student, you can take this objective starting with a sequence, identify the proteins or gene and the source. Here one test sequence has been taken, which is a protein sequence, identify the most probable protein and organism for this sequence. How can we perform this experiment, which can be seen? Okay. So, go to the NCBI querying sequence sites and go to the sequence program, and from that select the BLAST, and if you do not know how to go directly, then you can just use a search engine, and from the search engine box you can just type NCBI BLAST, and then from there directly you can go to that particular URL link of the BLAST. There exactly you can see the BLAST program. So, once again the BLAST main page will look like this, where the nucleotide, BLAST B, BLAST X, all kind of options are given. If we have an unknown protein sequence and we are seeking to identify by comparing it to the known protein sequence, so protein BLAST should be selected from the BLAST menu. Just click on this protein BLAST, it will open the BLAST P option. So, as in this problem, in this practical, we had taken a protein sequence. Once again we can take this protein sequence and directly paste in this entire search box, or either you can save this file in a FASTA file format and upload it. So, the BLAST P program is being used here and non redundant database is going to be used here, and the other parameters is a by default parameters can be set up, and the click on the BLAST options and this will give this output. The output will include all the parameters of by default values. So, here you can see the descriptions of the output result, where you can see the description of different types of sequences are there, then max score, total score, e value, save file options, accession ID, multiple sequence where options, and in the main menu options you can see the descriptions beside descriptions option, graphic summary, alignment and taxonomy options. So, this is how one can go through this one. So, in the score you can see the maximum score, which is the alignment score, and the total score, which is the all HSP scores are summed up, and the e value and the other parameters are also shown here. So, once you submit the protein sequence to the NCBI BLAST P options, the result will come and it can be shown here, where different options are there. The descriptions will show all the sequences which has been selected. As you can see here, all the sequences has been ticked one, and similarly there are a different options are available in this output result, which is just descriptions, scientific name, common name, tax ID, maximum score, total score, query coverage, e value, percent identity, accession length. So, user can uncheck this box. So, the output will be does not show the unchecked box in this output results. So, if user are interested to save a particular types of parameters, they can tick that one and that will be only revealed there. So, in the maximum score, it is a total HSP score if it is a 1 HSP in the alignment. If 1 HSP high scoring pair is there, it will be the my total score also, but if it is a 2 or more than 2, they will be summed up and that will give the total score. The E value we have already discussed in the earlier lecture that lower the value, it is more the significant. If the expected value is nothing but the result which is coming by chance. So, tending toward the 0, it is giving a significant meaning. The percent identity shows the total number of matches of the covering query sequence as well as the target sequence. The accession length tells about the length of the query sequence and the common length tells about the name of that particular sequences. So, these are the different types of options which can be seen. Similarly, in the next option is a graphic summary. So, in graphic summary you can see the alignment in the forms of color, where different color symbols are there and as per the alignment score it can be indicated. Here it is shown that all the alignment are in red color. The red color indicate that these alignments are showing a greater than 200 score matching. In this graphic summary, one can also see the conserved domain patterns of different color. So, in this graphic results, you can also see the conserved domain. Just click on that page, it will open a another windows. In this window, you can see the specific hits and non-specific hit. In specific hits, it is shown in a different colors, and in non-specific hits, it is shown in the different color. There is a black line which show the complete length corresponding to which the specific hit is made. So, one can go through this specific hit and they can find the detailed information about the hit made. What is this hit? We had already discussed in the earlier algorithm. There is a 3 steps are there in the BLAST database algorithms. One is a pre-processing of query sequence, second is a scanning of hits, and the third one is a extension of hit. In the scanning of hits, whenever the query sequence process, so this they make a word size, and whenever this word size goes and make a alignment with the database sequence, they are called a hits. So, from this one can identify which one is a specific hit, which one is a non-specific hit. So, coming again to the output of the BLAST result, there is another option called alignment options. So, in this alignment options, you can see there is a 2 sequences, query and subject can be seen which are aligned. So, wherever there is a exact match, the same character will be depicted there. There are options called downloads, genpeps. These options you can use, you can download this result at your local system. In this example, output of this alignment can be seen as a polymerase beta partial, which is a synthetic construct. So, these options also shows the different length in each column where the characters are matched. In the alignment, one can also view your results under the alignment tab next to the alignment view to select pairwise with dot for identities. So, in this slides you can see there is a no dot has been used. You can click on that options where the menu box option is there, where the dot option can be used, and wherever there is a match, the dot will be placed there. If there is a other than that, the other color symbols can be used there. So, this alignment will tells the exact pattern, where are the matchings of the characters are taking place, where are the differences in the characters are taking place, that can be easily identified. So, this is the one result. Similarly, if you scroll down these options, you can find out all the other options which are aligned in the sequences by scrolling down. One can download these options in a text file and that can be used for their later purposes. The other option is called taxonomy options. In the taxonomy options, you can see there is a reports in which the lineage, organisms and taxonomy, this is the sub options which can be seen here. Here in the lineage options, there is a column called organisms and then score, number of hits and the descriptions of that. Here there is a indications which has been highlighted in the red color, where one can see the organism's name Homo sapiens and their total scores and the number of hits. The highest number of hits has been shown in this slides, which is showing out of this total number of hits, it shows the highest number of hits, which is a Homo sapiens hits. So, what are the other options that can be seen with this output of results? There is a option of tree results or tree view which can be seen. The tree results can be obtained by a two-way, whether it is a rooted or unrooted tree, what kind of methods, whether it is a minimum evolution, maximum likely, neighbor joining, what kind of methods are there they are going to use for generation of the trees, that can be used here also. Here one can see the rooted trees here and all the sequences which has been used here, they are depicted and the different colors are indicating different group of the data sets. So, in this tree one can also see the tree methods, as I have discussed. In this method, fast minimum evolution tree method has been used. One can also see the maximum sequence differences. Here is a drop menu options, the user can select as per their need, but by default it is a 0.85 has been taken. The distance method which are used for this tree building is a Grissom protein sequences, by default has been taken. You can click menu options and the user can select as per their need also. Similarly, there are other options which is a multiple sequence alignment viewer option, which is a constraint based sequence alignment, and here the user can see the all the sequences which are aligned in a multiple sequence alignment point of view, and all of them are highlighted here in the red color. There are two columns, in the left column and right column you can see all the sequences name their ID, and that can be used for their further analysis. The user can manipulate the alignment by using a tools and other parameter which are available in this slides, and you can go on the web pages also, where you can tune the parameter, you can select the color options, you can save the file options, and other options are available, you can select as per your need. So, we have seen the different options of BLAST P output. So, finally, here what is the view? The description tabs to see a list of significant alignments. So, clicking on a protein's name, it displays the pairwise sequence alignments and links to the additional information about the protein and its associated genes, if it is available. So, you can see these options are there, you just click on the proteins, there the ticked one in a blue color, and it will show the alignment. Once again, here in this description you can see the maximum score, total score, E value, P value, and other options are there which can be used by the users. So, again the same alignment score you can see here, and in alignment you can see the pairwise sequence alignment of the query and database sequence, which we had already seen in the earlier slides. If there is a differences in the queries and databases, this can be highlighted in a red color. For the pairwise with dots for identifies displays, if any differences in the amino acid is displayed in the subject sequence, it will be displayed in the red color, as you can see highlighted in the red color of this alignment. Students, this is the comprehensive procedure of when one is going for the BLAST P options. So, here student we had taken this example of protein sequence alignment by using a BLAST P options. There are other options are available. I had not taken the other options, but as per the need the user can take the alignment programs. For examples, here I have taken only 2 alignment program, one is a BLAST N, the other one is a BLAST P. There is a other programs like Primer BLAST and IG BLAST and T BLAST X and other programs are there. So, you can use as per your need. Most of the times what happens, we do not know whenever the sequences are generated in the laboratory and we would like to find out whether this sequence is matching from which organism, then BLAST is one of the best program which you can use it. What you do here, whatever the sequences which are generated through the experiment in the laboratory, you save this sequence in a FASTA file format and you can upload to the NCBI BLAST database. From there, if it is showing similarity greater than 30% or more than that, it will be able to let you know the function of the BLAST program or the family of this sequence belong to which particular family, whether it is a gene sequence, whether it is a protein sequences. So, this way one can find about the function and characterization of the unknown sequence if it is given to the user. So, BLAST is one of the wonderful tools which is used for the users. So, Primer BLAST, as I discussed earlier also, it is very useful for designing the primer, which is a very good when we are going for the cloning program. So, and similarly there is a immunoglobulin BLAST, which is a very much useful one is going for the designing of immunoglobulin molecules. Mega BLAST is also a very important tool, one can use it when they are going for a high similarity sequence search program. So, thus this way the different versions of the BLAST is a very much useful for the user. Most of the times the users are selecting the programs directly on the web BLAST, but here again in the beginning of the slide I have discussed one can also download the setup of this software at their local system. The database size is quite big, the user can see their limitations of the storage capacity of their hard disk and accordingly they can save the databases at the local machine and then they can proceed for the local BLAST. One has to be very much careful about the memory size of their system and accordingly they can perform the alignment at the local system. So, students, we had seen about the standalone BLAST and also the web based. I have focused mainly on the web based program. It is quite useful for you. The user can execute this process from their own mobile phone as well as on the laptop or desktop computer. So, there is a vast range of the BLAST and programs are available. I am sure the student will be able to use this program very much as per their needs and it will be very much useful for their research work. Thank you very much student for listening me.